Uh, Hello, everybody. We'll get started. That's- Hello, everybody. We'll get started. So I'm very pleased to introduce Dr. Tammy Ryan today. She's that rare breed of being a very accomplished basic scientist and also an accomplished physician. She completed her undergraduate training at Western. She did her PhD here in Ottawa, and then a postdoc training at the Sanford, blah, 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 Previous Medical Discovery Institute in San Diego. Yes. She then went on to medical school at University of Toronto, completed internal medicine cardiology at McMaster, and she's now doing her fellowship in electrophysiology at the University of Calgary. She's been out of she's been in medicine now for 11 years, but she's kept her hand in basic science through her husband's lab. In fact, it sounds like her husband's been her postdoc for the last 11 years or something like that. So she's going to talk to us today about modeling cardiac disease lessons from stem cell and animal systems.
Thank you very much for the introduction and for the opportunity to speak to your group today. My only disclosure is that the transthyretin work that I'm going to talk about today is funded by Pfizer. With respect to what we're going to cover today, I'll spend a bit of time going over some basic models of cardiac disease and their relative contributions to the field. And then I'll talk a bit about modeling proteotoxicity, both in mice and in stem cells. And although this is a cardiology talk, we'll be spending a bit of time on Parkinson's disease because that's kind of where I got into this type of disease modeling. So I will subject you to a bit of neuroscience today. And then we'll talk about TTR cardiac amyloidosis, and we'll end on some theoretical plans for modeling of inherited arrhythmia syndromes using combined stem cell animal models. This was taken from a series of articles that were written a couple of years ago and published in Circulation Research on the growing importance of basic models of cardiovascular disease. And as you can imagine from this title, this person obviously thinks basic models of cardiac disease are important. Otherwise, they wouldn't be writing an introduction like this to this series. And yet there was a sentence in it that said, the road to failed therapies is littered with interventions that appeared promising in animal models. And I really took exception to this because I think the road to failed therapies is littered with a lot of things. Certainly some failed animal models, but I think there's also sometimes a lack of recognition of how much of what we do for patients every day has actually come from basic science labs and basic scientists. So statins, which are probably the most common drug that we all prescribe as cardiologists, came from basic scientists and were modeled in mice prior to being used. And more recent and sophisticated therapies like mavacamten for hypertrophic cardiomyopathy, and also gene editing, which I think is likely to make its way into the clinic in the next couple of years. Traditionally, basic scientists have used various models to study the heart in terms of both development and disease. The one that most people are probably familiar with is the mouse, but there are various other models that we've successfully used in these contexts. And actually, both the zebrafish and chick embryos have contributed a lot to what we know about cardiac development. In vitro models like HL1 cells, which are probably less commonly used, but certainly have made a significant contribution. And as a budding electrophysiologist, I've included xenopus oocytes here, which many of you might wonder why those are there. But actually, they're amongst the most common models used to look at ion channel function because they're just absolutely enormous, which makes patch clamping a lot easier than doing in cells like this. But there are certain contexts in which these models can't help us. And at the end of the day, with the exception perhaps of the immortalized cell lines, which have some limitations of their own, they're not human models. So the field of developmental biology was really upended in the late 90s, around 1998, with the advent of human embryonic stem cells. And this is Dr. James Thompson from the University of Wisconsin, who derived the first HES cell line. And this is a human embryonic stem cell colony here, grown on a mouse embryonic feeder layer, which is how we used to grow them. Some people still do. And you can imagine what a big deal this was for someone, a basic scientist, to end up on the cover of Time magazine. This was obviously a huge contribution that was recognized even outside of science and medicine. And interestingly, after these cells became available, people became interested, obviously, in using them to study differentiation from the pluripotent state to various tissue types in the body. And to do that, we had to develop protocols to try to actually do that with some efficiency. And for cardiomyocytes, that was really led by the work of Dr. Gordon Keller, who's still at UHN. And when I say led by him, I mean people all over the world were and are still using his protocols to differentiate stem cells, both iPS and embryonic stem cells, to cardiomyocytes. And how do we do this? Essentially, we copy what we know from developmental biology, and we expose the cells to the same signaling pathways that they would see during development to force them from the pluripotent state through to the mesodermal lineage, then to cardiac progenitors, and ultimately to cardiomyocytes. And certainly there are some limitations here. Those of you who are maybe more familiar with these models will know that limitations include they do tend to be a little bit electrophysiologically immature. But they do offer an unprecedented opportunity to study these processes in a human model. And this is around the time when I started working in these cells. These are some images taken from a paper that I published during my PhD when I was working here at the University of Ottawa under the supervision of Dr. Ilona Skarians. And we were interested in looking at the molecular mechanisms underlying some of these signaling pathways and how they drove cardiomyogenesis in these cells. Particularly, we were interested in Wnt signaling and how Wnt signaling impacted cardiac development, because Wnt is a bit weird in that we know early in development it promotes cardiomyogenesis, whereas later it flips to actually being inhibitory, and we were interested in that transition. And what I was able to show is that NKX2.5, which is an important cardiac transcription factor, interacts directly with the myosin phosphatase enzyme complex, and that's shown here by one of the subunits, MIPT1. This is a co-immunoprecipitation experiment. And that interaction is actually promoted by Wnt signaling through various mechanisms. And we were able to show that if you knock down expression of myosin phosphatase components, which is shown here by RNA interference for MIPT1, you actually prevent the ability of Wnt3A to inhibit cardiomyogenesis, and you get lots more cardiomyocytes, as shown here with myosin labeling. So a couple of years after embryonic stem cells were derived, the field was completely kind of turned on its head again by the advent of human-induced pluripotent stem cells. And I'll get into a little bit more detail of what these are, but really what they are for all intents and purposes is a homemade stem cell. So these are two iPS colonies here also growing on the feeder layer, and you can see that they look fairly similar, at least in gross morphology, to the human embryonic stem cells that I showed you before. And these cells were initially derived in the lab of Dr. Shinya Yamanaka in Japan. Again, a very big deal. So they didn't make it onto the cover of Time magazine like Dr. Thompson, but they were awarded the Nobel Prize in Medicine and Physiology in 2012. The prize was also given to Dr. John Gurdon because the work that Dr. Yamanaka did was really inspired by some cloning experiments that Dr. Gurdon had done at Cambridge many years before. So what are these cells and how do they work? How are they different from human embryonic stem cells? So what's different about them is that you take an adult patient, any patient, and you take either a skin biopsy or a blood sample or various other types of cells, and you essentially force them back to the stem cell state. And how you do that is by overexpressing the transcription factors that are important for the pluripotent state. And that is enough to actually reprogram them and convert them back to that. And once that you get them to that state, you can actually use them like a stem cell and differentiate them into any tissue type in the body. But the difference here is that because you've taken it from an adult patient, if that adult patient has a genetic disease, so does your stem cell. And so this really became what's known as the original kind of disease in a dish model, where people were not just now interested in studying development, but we could actually differentiate these cells into different tissue types and look at how a disease-causing mutation affected the function of those cells. And that's mostly what I'll be talking about today, which is disease modeling. Other interests for these cells are for cell therapy. So obviously, if you take a stem cell from somebody, they may be less likely to reject it in a cell therapy context. And I'm not going to talk about that today, but just to give you a sense, because I think I'd be remiss if I didn't. These cells are actively in clinical trials for macular degeneration, Parkinson's disease, and type 1 diabetes, amongst other things. So they are actively being investigated as clinical therapies. And then you can also use them for drug screening and discovery. And just to give you an example, mavacamten was actually modeled in cardiomyocytes derived from iPS cells before coming to market. I'm going to focus mostly on proteotoxicity today. And as I said, we're going to actually start by talking about Parkinson's, because that's kind of where I got into this and where the ideas for the cardiomyopathy transthyretin project came from. So since I'm talking to a cardiac audience, some of you may not be that familiar with the pathophysiology of Parkinson's disease. And if you aren't, PD is caused by neurodegeneration that occurs secondary to deposition of toxic protein aggregates in various organs, primarily in the brain. And these deposits are called Lewy bodies. And one of the main components of Lewy bodies is a protein called alpha-synuclein. So alpha-synuclein, despite having been heavily researched for many years, we know lots about alpha-synuclein. Nobody, I don't think, can say with any confidence that they know what the function of alpha-synuclein is. It also, at its baseline, exists in what we call an intrinsically disordered state. So it doesn't actually have much of a protein structure. It doesn't have any of the secondary or tertiary protein structure that most proteins will adopt. And for reasons that are really not well understood, all of a sudden, these proteins start to sort of stick together and form these oligomers, which then organize into fibrils and deposit into the cortex in the brain, the substantia nigra in the midbrain, and other organs, particularly in the gut. And then you get neurodegeneration as a result of this, as well as the other symptoms and signs of Parkinson's disease. And for many years, many different groups have modeled Parkinson's disease in mice using variants or mutations in alpha-synuclein, or in some of the other proteins that are important in PD, including Parkin, PINK1, or DJ1. And regardless of which of these proteins you mutate, these mice just don't have any significant degeneration compared to what we see in patients with these same variants. So in people, if you have a mutation in one of these genes, it's almost 100% penetrance to get PD. So you would imagine that amongst the many thousands of mice that have been put through the ringer in these experiments, you would see something significant, but in fact, they just don't show the same level of neurodegeneration. And this is true to some extent as well in other amyloid diseases, such as Alzheimer's. It's actually really hard to give a mouse amyloid, any type of amyloidosis. So we decided to try to model this in induced pluripotent stem cells carrying mutations that cause Parkinson's disease. But we actually were able to do this using an even stronger model, which I'll explain now, which is an isogenic HIPSC model. And what I mean by that is that in addition to having cells that have been taken from a patient with Parkinson's, which we can then differentiate to dopaminergic neurons, that mutation has actually been corrected using gene editing to create our control cell line or isogenic control, which we can then differentiate in tandem and has the exact same genetic background with the exception of the disease-causing mutation. And this has actually become kind of the standard in HIPSC modeling now in terms of the proper control to use. So we do this by differentiating the cells to DA neurons, and that's shown here. So from the pluripotent state through floor plate patterning, neural progenitors, and to fully differentiated dopaminergic neurons expressing tyrosine hydroxylase. This just gives you a sense for kind of the process that they go through. And we then looked in these cells to see if they had evidence of pathology that would be reflective of Parkinson's disease. And that's shown here in purple. So on the left here are two embryonic stem cell lines carrying mutations that cause Parkinson's in alpha-synuclein E46K and A53T. And you can see that they have these purple deposits within the neurites. And that's actually phosphorylated synuclein, which is one of the hallmarks of Lewy bodies that we see in human patients. Whereas their wild type counterpart controls don't really have much of this. This is also true in an iPS-derived neuron. So A53T, again, in synuclein, we see these deposits, and we don't see them in the corrected control. So these cells have some pathological features that we see in Parkinson's disease. When we look at the cells in terms of their structure and complexity, we see that they have much less in the way of neurite outgrowth. So the degree of arborization is much less complex in the A53T neuron relative to the corrected control. And we also see other deficits consistent with PD, including fragmentation of mitochondria. So that's shown here. So these are mitochondria labeled by MitoDS red in A53T human neurons. Compared to control, you can see that they're quite fragmented as opposed to this nice ribbon-like morphology that you see in the corrected. And that's also true in the A53T and E46K mutants as well. So having taken from this that we can see lots of different hallmarks of PD in these cell lines, we've also been able to show that you can actually transmit Parkinson's from a Parkinson's cell to a healthy cell. We thought, can we apply this model to a model of cardiac proteotoxicity? And specifically, can we use it to model transthyretin cardiac amyloidosis? So for those of you who may not be familiar with transthyretin, it's a protein that exists, it's produced in the liver. In its normal function, it exists as a tetramer, and it's secreted into the blood where it transports thyroid hormone and retinol binding protein. Just like synuclein, for reasons that are really not well understood, that monomer that tetramer becomes destabilized to monomers. Those monomers then become misfolded and then amalgamate into these oligomers, which then organize into fibrils and deposit into the heart, causing heart failure, as well as into the peripheral nervous system. So the neurological counterpart to ATTR cardiomyopathy, it used to be known as familial amyloid polyneuropathy, is now known as ATTR polyneuropathy. And this disease can be either inherited or sporadic. So the inherited form involves actual variants in TTR, and those patients have an early onset of disease that's often very aggressive, either primarily cardiac, neurological, or mixed. And interestingly, as I've discussed in a few of the meetings with people this morning, this disease went from being thought of as extremely rare, being very under-recognized, and having essentially no treatments, to having some of the most sophisticated treatments available on the market in medicine today. So we have RNA interference agents aimed at knocking down TTR expression. We have drugs that can stabilize the TTR tetramer, so preventing it from breaking apart. There are some amyloid antibodies that are currently being investigated, are not available clinically, but the goal is to try to actually mop up the existing amyloid deposits, though we're not actually sure if that's important, and we can talk about that if anybody has questions. Also, there's actually an ongoing clinical trial for gene editing of transthyretin to actually delete it out of the genome using CRISPR. So lots going on in this field, lots of interesting stuff. That being said, there are still some unanswered questions. And going back to sort of comparing mouse models to stem cell models as we did for PD, a couple of labs have tried to model transthyretin in mice and have had very little success. This was the most recent attempt that was published about 4 years ago. This group made a mouse model where they expressed human transthyretin in mice. And the levels of TTR in the mice were anywhere from the same level as humans, or 1x to 10 times the levels seen in humans. In spite of this, and having generated 3 different mouse lines, they looked at 59 mice. They saw amyloid deposits in 1 mouse out of 59, and that amyloid was not even TTR. It was actually another protein entirely. So again, keeping with the theme that it's very difficult to give mice amyloid diseases. And there's no mention in the paper of the mice having any phenotype whatsoever. So again, struggles in mouse models with amyloidosis. So then we started to think about how might we do this in stem cells to maybe answer some of the things that we don't understand about amyloidosis in the setting of TTR. And one thing that I didn't allude to in alpha-synuclein is that many people think that in amyloid diseases, the fibrils are what is toxic, and that's the assumption. But in fact, there's a fairly widely accepted hypothesis that the oligomers, the soluble oligomers in PD are actually what is toxic, not the fibrils. And that the fibrils might actually act as a sink to sequester the monomer, well, in this case, the monomers or oligomers away. And that therefore actually disrupting the fibrils might not actually help you that much. And so has been some work done on this in TTR, but much less so than in the neurodegenerative field. So the questions that we decided to ask were really, can we determine why TTR deposits in certain tissues and not others? And also, which of these species is actually toxic? And those are questions that we thought we could answer in a stem cell system, bearing in mind that there are some differences between TTR and alpha-synuclein that are really important when you're thinking about designing this model. So alpha-synuclein is produced within the cells in which it is toxic. So it doesn't have to go anywhere, although it can, we think, leave the cell and go to other cells and spread pathology. Whereas TTR is produced in a remote organ and secreted. And so when designing a cell-based model, you have to think about how you're going to mimic that. The other thing is that alpha-synuclein is deposited inside the cell, which is infinitely easier to model than TTR, which is deposited outside the cell. So these were things that we were thinking about. And so we thought, in order to answer these questions that I've told you are of interest to us, the two things that we really need are a representative model that mimics what we see in humans, and we need TTR protein. And obviously, we were not the first ones to think about doing this. It had been attempted in various different cell lines, not in stem cells, but in various immortalized cardiac and neuron cell lines. But actually, compared to other amyloid diseases, the amount of literature in this field was like infinitely smaller. And that really should have been kind of a red flag before we started this project. But the take-home message was that from the little work that was available here, likely people thought the monomers and the oligomers were going to be the toxic species more so than the fibrils, in keeping with what we know about alpha-synuclein. So we thought, okay, well, you know, there's very little work done here. We're really good at this. We've done this in Parkinson's disease. We've published lots of good papers, lots of good data. We're going to come in and we're going to do this with TTR, and it's going to be great. Turns out it's actually extremely difficult. So TTR is a very difficult protein to work with relative to alpha-synuclein. So if you put alpha-synuclein in a tube and shake it for 2 days, it will make fibrils. Not so for TTR. So this is an experiment that one of the grad students in the lab did, where he actually tested 60 different conditions to try to generate TTR fibrils. And you can see that only 2 out of the 60 conditions actually did that. And he did this by varying the amount of TTR that he included, varying the buffer conditions, varying the amount of seed, which I'll get into. And ultimately, only 2 out of these 60 conditions actually generated fibrils, which gives you a sense for how challenging this has been. We also use dynamic light scattering to look at, to make sure that we in fact have different structures in these reactions. So this is a technique that looks at essentially the distortion of solution around a molecule to give you some insight into its structure. So we have monomers here shown in green and the tetramer, which overlaps because they adopt a certain structure. And then you can see that in those 2 conditions, we start to see other structures shown here in the 2 variations of blue. And those we actually think are oligomers. And in purple, we have fibrils that essentially we have purified and given as sort of a control. So then we looked at these under transmission electron microscopy. And we have these little kind of blobby things that we think are likely oligomers, and then these longer species that we think that are representative of fibrils. So now we have our transthyretin. We're able to make various structures of TTR, so that's good. But then we thought, how are we actually going to put these on cells and have it be reflective of disease? Because as I've told you, it deposits outside the cell, and so just putting it onto cardiomyocytes probably doesn't make a ton of sense and is probably not reflective of what actually happens in patients. So my thinking was that if TTR is depositing outside the cell, there's probably going to be some interaction with the extracellular matrix. And indeed, we know that TTR can interact directly with perlecan, which is a common component of the cardiac ECM, and that patients with transthyretin amyloidosis actually have increasing expression levels of ECM components. And these are just images, electron micrograph images of fibrils taken from patients at autopsy. And you can see that they basically just completely take over the tissue until the cardiomyocytes are, for all intents and purposes, wiped out. So we thought we'll try and mimic this by actually providing an extracellular matrix similar to what the cells might see in vivo. And so we did this by setting up a co-culture system where we use cardiomyocytes along with cardiac fibroblasts. We put them, and these are derived from iPS cells. We put them into culture and we allow the cardiac fibroblast to secrete an extracellular matrix, which we hope then mimics, at least in 2D, what might actually occur in the heart. And then the goal is to use this and actually treat it with the different forms of transthyretin to see what happens. So this just shows you what our cardiomyocyte alone system looks like. So we have a GFP reporter under the control of a cardiac progenitor gene. So at day 0, the cells are not green. They grow in these colonies and they express markers of pluripotency. By day 9, once they're cardiac progenitors, they start to turn green and express NKX, which is a progenitor marker. And by day 17, they're actually beating in culture. We then take these cells, so these are the same cells here labeled with myosin, and combine them with iPS-derived cardiac fibroblasts, which are shown here by expression of vimentin, to make this co-culture model, and TD7 is also a cardiac fibroblast marker. I should say that we do have some cardiac fibroblasts in our cardiac cardiomyocyte differentiation, and that's shown here by labeling of TD7 in red, but not enough that we thought would secrete an extracellular matrix that was actually useful. So that's why we supplement it with additional cells. To give you a sense of what this looks like in terms of the actual ECM that is produced by these cells. So these are the cells in culture labeled with laminin and collagen. And this is a slice through the culture, and you can see how extensive the network outside of the cells is. And so we think actually that this probably is a pretty decent ECM to work with in this context. So we really just kind of started these experiments, and the data I'm going to show you over the next 2 or 3 slides are quite preliminary. So we treated these cells with both wild type and monomeric TTR. And what we found was actually kind of surprising. So we treat them and then we, wash it off and fix the cells. And the cells that were treated with wild type TTR, we don't actually see much TTR left behind after we do this. But when we treat with the monomeric TTR, we actually see a ton of TTR deposition in these cultures. And we thought, okay, well, that's probably because we know from these previous studies that the monomeric TTR is toxic. And so we're probably going to see some toxicity with this. But in fact, that was not the case. So this is a quantification of deposition. So the mean volume of TTR as a function of the volume of myosin. And you can see that if we treat with monomeric TTR at a concentration of 20 micromolar, we get a lot of deposition, not much so in the wild type. And to give you a sense, the physiologic concentration of transthyretin is about 3 to 7 micromolar. So that's somewhere in this range. We don't know what the actual toxic concentration is. So I showed you the image of those fibrils. I mean, that concentration is going to be enormous. And so we've started on the lower end, and I'm sure it's actually much higher than this in real life. If we look at toxicity using gamma H2AX as a marker, so it's a marker of double-stranded DNA breaks, in the presence of monomeric 20, which is depositing at huge amounts, we actually don't see a significant increase in the gamma H2AX in spite of that huge amount of TTR being deposited. We actually did see some in the wild type, which was pretty weird. And so then we thought, and as I said, this is preliminary, so this is something that we're working on now. Is it that deposition of monomeric is actually protective? So the monomeric deposits in the ECM and therefore is not able to exert any toxicity on the cells? Or is it that that deposition is actually the initiating event for oligomer and fibril formation? And if we took this out longer, we might actually see toxicity if those fibril or if those monomeric proteins were actually able to interact and form higher order species that might be toxic. So those are the next questions that we're hoping to answer. So take home messages from this part. So we've designed a biomimetic model of ATTR cardiomyopathy. We've shown that monomeric TTR is preferentially trapped by ECM derived by iPSC cardiac fibroblasts. And then we're looking to increase the complexity of this model to better understand this. So We want to understand what triggers the instability that causes TTR tetramers to fall apart initially. We're planning to manipulate ECM components using CRISPR-mediated gene editing. We also want to include TTR purified from patient plasma. And the reason for this is you might say, well, you showed us all these images saying you can make oligomers and you can make fibrils, but you didn't show us any images of those actually being put on cells. And the truth is that the yield of this is extremely, extremely low. And in our work and in work with collaborators, what we found is that you actually, like what the group who made the mouse model showed, you actually need a little bit of patient material to get these higher order structures to form. And so we need to be able to figure out how to do this to an extent that gives us enough material to actually be able to do these experiments. And that's what we're working on now. In terms of preliminary thoughts around what causes the instability, we actually did some modeling using AlphaFold, which is an artificial intelligence program offered by Google. It's actually extremely good at predicting protein structure. And what we were able to show is that if you successfully remove parts of the N-terminus of the protein, it actually results in instability. And why is that important? So people have shown that in patient material, in patients with ATTR cardiomyopathy, the fibrils actually contain transthyretin that's missing a little bit of its N-terminus. And so there's been some thought that maybe cleavage of that N-terminus is what actually precipitates destabilization and fibril formation. And so that's what I'm showing you here. So this is a TTR tetramer with just the first 4 residues of the N-terminus cleaved off, then the first 9 residues, and you can see that it still maintains a decent structure, and the confidence of that structure is high, as shown here in blue. But as you start taking away more and more amino acids from the N-terminus of the protein, the confidence of the structure is lost. And you can see here, you don't have to be a crystallographer or a protein structure expert to say that you've lost your interaction. And so that's something we're interested in exploring further. We've also started purifying this from patients, which was also a struggle, but we think we have working pretty well. So this is a fractionation experiment from patient blood, patient with ATTR cardiomyopathy. So you can see that in the insoluble fraction, we're able to detect multimeric species of transthyretin, which are not present in the soluble fraction. We can actually purify these using ion exchange chromatography and detect multimeric TTR from that. And so this is what we're now using to try to generate higher order structures, which we can then use to better model toxicity. We're also currently working with Dr. Noel Fine at the University of Calgary, and also Dr. Patterson here is a part of this group to generate a biorepository from these samples, which would then be linked to the Canadian Registry for Amyloidosis Research, which is a pre-existing registry that tracks these patients. So that's everything I wanted to tell you about TTR. And the last part of the talk, I wanted to just cover what I would like to move into doing in the next couple of years. And so there won't be any real data in this section. It's more of my interest moving forward. And that's in modeling of inherited arrhythmia syndromes using iPS-derived cardiomyocytes as well as animal models. In case anybody's not familiar with this group of diseases, there's used to be 4, more recently now 5 conditions. So long QT, short QT syndrome, Brugada syndrome, catecholaminergic polymorphic ventricular tachycardia, and the new condition that was actually really fully characterized here in Canada called calcium release deficiency syndrome. And these diseases are extremely rare, but they also manifest as sudden cardiac death, often in young patients. So while they're very uncommon, the clinical consequences of them are just about as bad as you can imagine. And so over the years, we've learned a fair bit about these, particularly about long QT, but there's still a lot about these conditions that we really don't understand. And I'll talk mostly about long QT and Brugada syndrome today, but a lot of what I'm going to say would also apply to the others. But I think these, in terms of explaining sort of what I have in mind, these are the appropriate prototypes. So long QT syndrome, there are various different types of it. 3 of the most common, long QT 1, 2, and 3, are caused by loss of function variants in potassium channels or gain of function variants in sodium channels. And that results in either loss of outward potassium current or sustained inward sodium current, such that these patients develop repolarization abnormalities. After depolarizations, develop arrhythmia, and they can die from those arrhythmias. Brugada syndrome, also to some extent caused by loss of function, though, in sodium channels. But we have a much, not nearly as good of an understanding of the genetics of Brugada as we do of long QT. So we only really detect variants in SCN5A in about 20% of patients with Brugada, and the rest remain without an identified variant. And the hypothesis is that for the most part, these patients probably have a polygenic condition rather than a rare monogenic culprit. Modeling these conditions in animals has been challenging because essentially the action potential in mice is different than in humans, because the channels that they rely on and the currents that they rely on are to some extent different. So on the left are shown the inward currents, and on top you have the human action potential and the mouse action potential. And on the right are the outward currents. And in red, the authors have circled the currents that are different between humans and mice. And you can see on the inward side, things are relatively kind of the same. But on the outward side, there's a significant difference in the channels that mice used for repolarization as opposed to humans. In humans, the major repolarizing currents are IKS and IKR, the inward rectifier channels. And these are two of the channels that are affected by mutations that cause long QT. Whereas mice rely primarily on ITO, or the transient outward modifier, and IKS to a lesser extent. So the result of that, in addition to having a different action potential shape, mice have a completely different QRS morphology on ECG. And you certainly can cause mutations in these and cause a long QT in mice, but the most that they really get is non-sustained ventricular tachycardia, and they very rarely die from tachyarrhythmias in general. And for perspective, the baseline heart rate of a mouse can be 500, and the ventricular tachycardia that they're getting is about 550. So it's just not really, the mouse just goes on about its business, essentially in VT. So I've been thinking for the last little while about how iPS cells might be able to contribute to this. And certainly there's been some work done by various groups around the world to try to better understand these conditions in the human context. And I wrote a review about this with Jason Roberts that was published last year. Interestingly, long QT was actually one of the first diseases that was modeled in iPS cells. So this paper is 15 years old now. This came out very shortly after the paper that actually derived iPS cells. And this group was able to show that a lot of the things that we knew about long QT from animal models, from patch clamping oocytes, also held true in iPS cells. But people have since built on that to show that there are other mechanisms, and also there's an ongoing clinical trial. So I'll just walk you through this. So this is a figure showing the molecular mechanisms of long QT as shown in iPS-derived cardiomyocytes. And you can see that primarily one of the main issues is channel trafficking defects. So the channels are misfolded, and either as a result of that get stuck in the endoplasmic reticulum or in the Golgi, and they never make it to the plasma membrane. And so that's why essentially they have these abnormalities. But in addition to that, there are differences in the stability of the mRNAs translated or transcribed from these genes. And people have really been able to use these mechanisms to kind of think about how might we treat this in a more targeted way than a beta blocker, which is what we generally give these patients. And this is a particularly interesting example, I think. So lumacaftor is a drug that is related to the drug that is used to stabilize channels for transport to the plasma membrane in cystic fibrosis. And so the goal of this was really to see, can we rescue transport of the channel to the plasma membrane by restoring folding of the channel to its proper structure. And in iPS cell model, they actually were able to do that. And this drug is currently in clinical trials for long QT treatment. Brugada, on the other hand, is a little bit of a mess. So these are data taken from a Nature Genetics GWAS of Brugada syndrome. And you can see SCN5A is the most common gene that comes out. But there's at least 10 other genes that contribute to Brugada risk, likely in a polygenic setting. This is very difficult to model in the mouse, if not impossible. It's really hard to give a mouse this complex genetic background Whereas iPS cells are actually ideally suited to this, because if this is the patient's genetic background, that's going to be the genetic background of the cardiomyocyte you're going to get from that cell. So then you might think, well, you know, we'll just model all Brugada and iPS-derived cardiomyocytes, and we'll figure out all about Brugada, and we won't have to do any other experiments. And this is a figure from that same review showing all the mechanisms of Brugada that have been shown in iPS-derived cardiomyocytes. And you might look at this and say, well, there's just as much of this on this slide as there was on the long QT. And so, you know, we must have learned a lot about it. But in fact, a lot of these pathways have only been shown in one lab, have never been reproduced, and are from iPS cell lines that were made carrying variants that are extremely rare, maybe in one family in the whole world. And so are not sort of informative on a larger scale of Brugada mechanism, at least in my opinion. And so to give you an idea of what the landscape currently looks like, these are all the iPS models of Brugada syndrome that have currently been generated. And you can see that a few of them have mutations in SCN5A, but some of them are in other genes that are not known monogenic causes of Brugada syndrome, and almost certainly are not. And actually, most of these variants are of uncertain significance. There's only two that are pathogenic, one pathogenic, two likely pathogenic. And so this, I think, presents an important disconnect between what's happening in the lab versus what's happening in patients. And really, I think one of the take homes from this is that we should probably be focusing, you know, to some extent more on the variants that we know are important in Brugada. But then the flip side of that is, well, you know, if we know that this is polygenic, why don't we just take these iPS cells from these patients without variants, make cardiomyocytes from them, and model Brugada syndrome? And some people have already tried to do that, and turns out there's nothing wrong with the cells. So this is a paper from the group of Connie Bazina, who's been very important in the inherited arrhythmia field. And they made, I think there were 4 lines in this paper from patients with clear Brugada syndrome, no variant identified in SCN5A. And they put them through a battery of electrophysiological tests and could not find anything wrong with them. The cells were, for all intents and purposes, completely normal, even though we know these patients have had sudden cardiac events. And so clearly the cells are not normal. Which brings me to the complexity of Brugada syndrome. So I'm not going to go into this in any detail in the interest of time, but just to sort of walk you through this, there are multiple hypotheses sort of that different people defend about how Brugada syndrome is caused. Initially, this was thought to be a pure channelopathy, but we know now that there's almost definitely a structural component to this. And so there's the depolarization hypothesis and the repolarization hypothesis, which, you know, there are 2 camps that kind of defend one or the other. But I think what you can take from both of them is that in some way there's a gradient of electrical activity that's being generated that is then contributing to arrhythmia. And so that suggests an underlying structural issue. And then the third hypothesis, which is likely not mutually exclusive to these 2, is that migration of neural crest cells, which contribute significantly to the right ventricular outflow tract, which is thought to be the site for some of these gradients, may also underlie the pathogenesis of Brugada syndrome. So then we come to the depressing realization that we have a polygenic substrate, which we cannot really model in mice. And we have a structural substrate, which we cannot really model in iPS cells. And certainly, if anybody here is familiar with the iPS literature, you might say, well, you know, there are, you know, cardiac organoids available, but I just don't think that those are kind of at the point yet where we can model something like this in that system. And so then the question that I'm hoping to ask is, what if we combine the animal and stem cell models to try to answer this question? And that would be done using a technique called blastocyst complementation, which some of you may be familiar with. So what this involves is taking stem cells and injecting them into a developing embryo at the blastocyst stage, such that the animal that you end up with at the end is composed to some extent from the original animal cells, but also from tissues generated from the stem cells that you injected during development, so that you end up with a chimera. Obviously, there are some ethical challenges with this because, you know, if you're talking about injecting human stem cells into a mouse, then, you know, you've generated a mouse essentially with human neurons, and there are some significant ethical questions around that. such that people have started maybe working a bit more with progenitors to kind of avoid generating mice with human brains, basically. But this has been done for cardiac and the vascular system in mice using stem cells, so it is theoretically doable. And so the idea here in terms of what I had in mind, and I've shown it here for Brugada syndrome, is that you would have a Brugada iPSC, which you would then inject into a mouse blastocyst, generating a human mouse chimera that has essentially a heart that is structurally mouse, and so has all the anatomic substrate required, but has the polygenic basis of the human Brugada patient. And the idea here would be that you could model the neural crest contribution to this, both through tracking migration of the cells and contributions to the RVOT, again, by tracking and also by single cell sequencing. You could also likely track the etiology of the fibrosis that we know we see on MRI in Brugada patients, and ideally interrogate the basis for arrhythmogenesis in these patients. So in summary, inherited arrhythmias have a complex genetic and structural substrate that are not well suited to traditional disease models. And I think a lot of what I've said would also apply to CPVT for those of you who are interested in inherited arrhythmias. And the answer to this may potentially be the generation of chimeric mouse models that rely on both the structure of the mouse organ and cardiomyocytes derived from stem cells to provide a solution to this. So just to acknowledge, so the group at the University of Calgary, so Dr. Scott Ryan, and a lot of this work was done by Shivani and Gerardo in the lab. Dr. Noel Fine, who's our collaborator on the patient side for cardiac amyloid. And then Dr. Lorena Sandices at UT Southwestern has provided us with patient material to test the role of seeds, as has Dr. Lorraine Connors in Boston. So we're really good. Thank you.
It's really super. I understood about 10%. So I'll open questions online. Here's
Hi, Tammy. Katie Rayner. Well, we're going to meet in a couple of minutes, but that was fascinating. So thinking about your TTR stuff, one of the things that immediately comes to mind when you're describing this extracellular deposition and the differences between human and mouse and all these other models is this idea of modifications that proteins go through when they're being just cycled in the cell, and how we know, you'd think human and mouse would probably be similar, but what's known about that? Just the natural, as the cell is, the TTR is being made, other, whether it be sugar or phospho or other modifications, and have you, explored any of those?
We haven't explored them, so not much is known. Certainly it's phosphorylated for sure. Not much is known about it, but we have talked about this, and one of the ways that we thought maybe potentially get around this, and it again adds to the complexity, but I think would address that, is actually, rather than making recombinant TTR, is actually to make liver cells, which then secrete the TTR.
Exactly.
And then the TTR comes out looking the way it should.
Yeah.
And so we have thought about that. It would be something new for us because we've never made liver before. We certainly can. And I think that's probably the easiest way around that. And then you're talking about making liver, secreting the TTR, purifying the TTR, and then exposing it to the seeds.
Yeah.
So it gets complicated, but I think it's important because I think that's why, we haven't been able to understand this, because in the previous studies, people are just kind of throwing recombinant TTR on there, and you're losing that complexity that I think is really critical.
Exactly. And I think the world of recombinant proteins, right, learned this through bacterial expression systems, plant expression systems, insect, human, so I think there's certainly room where it's not ideal. You can't produce maybe the same buckets worth that you did, that you do from bacterial produced systems. But the other thing is, when you were showing the alpha fold and you were showing with the N terminus that looks like it might be cleaved, I mean, that's a bit of a chicken and egg too, because sometimes if it's unstable, it's hard to detect depending on how, so, it may not be cleaved. It could just be the stability of the protein itself. And then the, AlphaFold is taking that information also and then not able to accurately predict it based on the, so there's sort of like you can kind of sometimes get in a bit of a circular argument when you look at, but again, maybe there's an endogenous cleavage or instability or modification that happens at that N terminus.
Yeah,
that you might be able to detect just by like simple mass spec, just from your liver, if you're able to produce it in liver cells.
Yeah, so to that point, so when there is actually a cryo-EM structure, in the cryo-EM structure, that N terminus is absent.
Okay.
In patients, but it comes back to exactly what you said. In cryo-EM, if it's mobile,
you're
not going to see it, right? And so the original data saying that N terminus might be missing is mass spec from patients.
Okay.
So we do think that piece is missing, but you're right. At what stage that actually happens, we don't know. And yeah, ultimately, I think the other, and going back to your thing about the modification, this is also why we want to try to get a lot better at purifying this from the patients.
Right.
Because I think there's actually, maybe I'll just show you the slide. I didn't include it because I didn't want to go over, but so this is actually a paper from Science Translational Medicine that was published about 7 years ago. So they actually looked at what was circulating in these patients using a probe that they designed to only detect oligomers. And so when they looked in patients with ATTR polyneuropathy, they were able to see these multimeric structures floating around, and this is a mutation that causes ATTR polyneuropathy that they didn't see in healthy donors. And interestingly, they also didn't see in patients who were asymptomatic. But then when they went and looked in people with cardiomyopathy from ATTR using the same probes, but obviously a different mutant, they couldn't detect any circulating oligomers. But I showed you in that Western that there are circulating oligomers. And so we think that even between the two diseases, the structure of the proteins is probably not the same, because if you're using the same probe and yet you can't detect the same structure in a different patient, I think there's a lot going on here. And so I think being able to purify this in higher yields from patients where we can actually look at this a little clearer, I think is really critical.
Hello, Dr. Ryan. Thank you for your amazing talk. I really enjoyed it. So I have two questions. So first, is about the last part. You mentioned we could generate chimeric mice that were born with a mouse heart, but with a human cardiomyocytes. But my concerns is this mice probably they would not survive because the human heart in a healthy person, the heart rate 70, 100, right? If is maximum probably around 150, that's the limit because of the long action potential of the human cardiomyocytes. The mouse, they can reach up to 600 because their actual potential is so short. So if we generate a mouse with the human cardiomyocytes, so the maximum heart rate, I would say would be around 150 or 200 maximum. So probably it won't be sufficient to support the cardiac output.
You might be right. And I have thought about that too. So they have been generated between mouse and rat and mouse and human, but not taken to like a very, not taken out very far to really understand that component of it, but you might be right. I think one thing that might save it is, you're not going to get 100% human cardiomyocyte contribution to the heart, but then the flip side to that argument is, well, then, are you going to get arrhythmogenesis because you have, some cells that have action potential that's way longer than other cells that are way shorter and want to be way faster. So I think it's a technology and evolution for sure. And you may be right. I think that's going to be a significant challenge. But I think it's still worth the try.
Yeah. Okay. If you, if the heart is mostly mouse cardiomyocytes, like the LV, RV, only the RV flow out tract, has the human cardiomyocytes, I mean, it may have a better chance to work.
Yeah, and I think you could
just
also
some thoughts.
Yeah, you could also try to design a system for that specific purpose, where the neural crest population only was what came from humans. I think there's going to be some nuance to it. I agree.
Okay. Thank you. Second question is about the TTR, the ECM. So when we do cell culture in vivo studies, it is important that we control the condition so that the mimic the in vivo condition as much as possible. In studies of ECM, it's important to control the cell density. So in the in vivo heart, or the ECM, they are surrounding the cardiomyocytes. But in the cell culture, most of the ECM, they were separated underneath the cell layers. I'm wondering if the ECM that you measured or imaged were they mostly underneath the cell layer, or they are wrapping the cells from the or other directions?
Yeah, that's a good point. I don't think any of the imaging that we've done would clearly answer that. I suspect you're right, and a lot of it probably is kind of sitting underneath rather than kind of infiltrating in between. But I don't think we've done imaging that's good enough to say that for sure.
Okay, so do we know how the TTR can promote the ECM deposition? Doesn't, let's say, affect the cardiomyocytes so that they die to free the space for the ECM to deposit, or they just stimulate the fibroblasts to secrete more proteins?
I suspect it's something similar to other paradigms where you see increased fibrosis. It's essentially, I think it's a stress response. So in many different conditions, cardiac fibroblasts get activated in response to stress, and one of their responses is to secrete more ECM. I suspect it's a general stress response that, like what we see in those other conditions. That would be my thought, but I don't think we know that for sure. But that seems to be the simplest, probably, explanation.
Okay. Thank you so much.
Thanks.
I was very intrigued by your observations on Brugada syndrome. I didn't realize that it was such a, 20% are now thought to be monogenic and 80% are polygenic. Is there a sense that they're different diseases? Do they behave different clinically? Yeah.
I don't know that they behave. I don't know that we know enough about it to sort of put them into boxes. Certainly, there are some, overlap syndromes that have kind of been described, and I kind of showed it on that slide there, where people have thought, well, this PKP2 variant is showing a Brugada-like phenotype, so maybe PKP2 is a causative gene in Brugada in addition to ARVC. That ended up not being true. There are some patients who have variants in TBX5, which did show up in that GWAS. And so I think one term I've seen used that I think characterizes it well is that maybe it's a spectrum within which, it's a similar disease, but maybe slightly different depending on what your polygenic profile is. But I don't think that we understand it well enough to say, there are these 5 boxes within which Brugada patients fit, and you go into this box. But I think that is the goal of the polygenic risk score, to ultimately do that. And so that you can actually tell a patient, your risk is greater or lesser because of your polygenic profile. And certainly I think that's how Jason Roberts thinks about it, when he's building these. But we're not quite there yet.
That was a fantastic presentation. Thank you. I think you hit the nail on the head. I think Brugada we always saw as this channelopathy, and then post-mortem studies have clearly shown people have these structural abnormalities. And I think there are some hypotheses going around about how these gap channel dysfunctions through connexin 43 leads to this structural change. So I don't think it's a fully, I don't think we've fully explored or understood that concept. And I think there's more work to be done. There are some groups as well trying to draw more towards the arrhythmogenic cardiomyopathy side to say, actually, maybe Brugada is a structural disease. Maybe it's on a continuum with other forms of arrhythmogenic cardiomyopathy. it's not obviously a great model for a monogenic disease because clearly it's not in most cases. The thing I was going to raise really was, one of the challenges we see in inherited arrhythmia are these families where clearly there's an increased risk of sudden cardiac death. And we do a full phenotype, we don't find an obvious cause, but we know there's something there. And the question is, how do we go about exploring those families? I know Wayne Chen has done a lot. CRDS, was a great Canadian story for discovery, but how we look at these families and what we could be doing more to try and understand these sorts of diseases.
Yeah, I think that's a really great point. And like I'm like a CRDS convert. Like I think that story is so cool, the way they went just from mouse model to fully essentially characterized disease to now we're modeling a diagnostic test. And this all happened within like 15 years. And basically through collaboration of basic scientists and clinicians, like really incredible success story. I think I wish there was more of that, because I think there's potential that there are, maybe not a ton of, conditions specifically like CRDS where we have a clear culprit and they're going to end up being as sort of not monogenic, but you know what I'm kind of trying to say. Like, I think there's more to be done there, as you said, because there is a huge proportion of these patients who never really get an answer for, what's happened to them. And do I think iPS cells have a role there? For sure, because I think, those patients almost certainly have some polygenic substrate. I think we've probably reached the point where there's very few rare variants, monogenic causes of these things that we have yet to identify. I wouldn't say there's none, but I'd say there's probably pretty few. And so I think these people are polygenic, and I think this is where iPS cells kind of shine, because that genetic background, you just can't do that in an animal model. So I think lots of work to be done.
Right, time. So thanks again. And if anybody wants to ask Tammy any questions, she's around for the rest of the afternoon. So.